All right, so our next speaker is Michael Sikora, who is here at Stanford University. He works in Lars Steinmetz's group in the Department of Genetics, and they have been collaborating with Ron's group here for about a year now. Recently, we're awarded an R01 to work on from the NIH to work on this project exploring the role of T cells in ME/CFS.
Well, thank you. I invited Michael here to give you a presentation, and Michael is a graduate student. And why I'm doing that is for you to see the quality of students that come to Stanford. And these are the people who make a difference in the research. And to help him in his career path, I said, you're in charge. And so you have to herd these three faculty members, Mark Davis and Lars Steinmetz. Okay.
It wasn't you. It was...
and me, and tell us what we have to do. And so he's taken on the challenge, and I think this is really good for his career. And so this is just an opportunity to see what a student is like, and you not have to hear everything from the people who actually don't do the experiments. Thank you, Michael.
Yeah, thank you, Ron. I hope now that you've put me in charge, I can stop doing the experiments, actually. But really, it's an honor to be up here amongst so many really established researchers. And so today I'm going to be talking about a project that we have developed in the lab that we are now applying in the context of CFS. And so I tried to tone down the science to make it approachable for everyone. It may still be a little bit dense, but I will alert you guys to the kind of important points that I think, at least for preliminary results, are promising. And so the project that I'm going to be talking about today is about the immune system. And of course, there's been a lot of talk that the immune system may be heavily involved in CFS. And so, as you may know, there are many different pathogens that can infect humans. These are from viruses, from bacteria. Ron also mentioned like fungal. And so, for example, the flu and herpes are all viral infections, and then you can get tuberculosis or pneumonia from bacteria. And so what you can see from this slide, at least, is, this is only a fraction of all the organisms that can infect people, but there are a lot of them, and they can happen all throughout the body. And so the immune system is tasked with actually detecting these infections and neutralizing them. And so this is just a figure showing a pathway that the immune system kind of travels through in order to detect all of these potential invading organisms. And so today I'm going to be talking about T cells specifically. This is a particular cell type within the immune system. The immune system is very complex. And so this is somewhat narrow, but just to let you know kind of how these T cells work. So they go throughout the body through this lymphatic system. They're also in the blood. And so the T cells go all throughout the body and search for all of your cells. So these are, heart cells, brain cells, skin cells, etc. And they have this T cell receptor here. It's kind of like an arm on the T cell that feels around and looks at the cells like indicated here, which may be infected or may not. And so based on a very specific interaction of this T cell receptor with this cell, it will be able to determine whether the cell is infected, and then if it decides that it is infected, it will then kill this infected cell. And so, as I mentioned, there are many potential pathogens, and so whatever bit in red here is kind of presented to the T cell, that is very variable. And so the immune system has to create a bunch of different T cell receptors. And so I won't go into how that is done, but basically you have many, many T cells throughout the body, and each one has its own T cell receptor. And so that way, when you have any different type of infection, each T cell with its own T cell receptor can then recognize such an infection and remove whatever invades the body. So you start with many T cells, each one with its own T cell receptor, which you can see as indicated by colors. So each one has its own specificity. And upon infection, some of these T cells will recognize an infected cell, and it will undergo a process known as clonal expansion. And so this is a critical point in this talk. So this clonal expansion, when this T cell recognizes the infection, it will duplicate itself and maintaining that same T cell receptor. And so in that way, now the immune system has a bunch of these blue T cells that all recognize these infected cells, and so you can create a stronger response to the infection. And a very key point here is this interaction with the T cell and the infected cell, which I mentioned before. It has to be very specific. And so this clonal expansion won't occur, just randomly. It has to have something that it recognizes in order to clonally expand. And so that may have been a bit of background, so I'll just summarize what I've said so far. So the immune system is in charge of searching the entire body for infections. The immune system, and in this case T cells, will kill these infected cells. Each of these T cells have T cell receptors that are very specific for a particular infection. And if it is specific and recognizes the infection, it will clonally expand. And it's rare that any one T cell will recognize a particular infection. And so this is the first data slide that I'll present. I believe Mark Davis talked here last year and showed this slide, but so this is kind of the crux of the project that we are going to do, which I'll talk about in the second part of my talk. So each circle is a pie chart, and this represents the diversity of T cell receptors in an individual. And so on the top, you can see are the healthy controls. Most of this pie chart is white, indicating the T cells they all have a unique T cell receptor, which means that there's been very little clonal expansion in these healthy controls. However, if you look at here is Lyme patients, and at the bottom is MS patients, which have been mentioned, multiple sclerosis. These are well-known immunological diseases. There is a T cell response to these diseases with a known infection. And so also you can see here are the CFS patients, and they have a similar pattern. So this suggests that there is at least an immune response in CFS patients, which you do not see in healthy controls, and you do see in other diseases that are known to be to have activated immune responses. And so just to go back to the cartoon, in CFS patients and these other diseases, you have this, a lot of clonal expansion. The healthy controls just have these unique T cells. Okay, so that is kind of the background and why we are starting our project looking at these clonally expanded T cells. So before I get into those results, I will give you some more background. And so this is, the fundamental slide of background. It is known as the central dogma of biology. So basically, I think most people are aware that we have DNA in our cells. These have genes, which, in your genes, they make RNA, which go on to make protein, and these proteins are functional, and they go out and do things like kill infected cells. So we use a technology known as RNA Seek, and so basically what we're doing is reading the RNA in cells in our body. So I'll walk you through that as well. So here, if you think of this line as your DNA, there are genes all throughout your DNA, indicated in red. These genes, from time to time, will be expressed, and so they will make RNA, and this RNA will go on to make protein. And so the basis for our project is the fact that RNA recapitulates the protein, which is what is going out and enforcing its function. And so you can imagine these different genes have different functions. If you take heart cells and do RNA-seq, the genes that are important for heart function will be expressed. Likewise, if you do it in kidney, the kidney genes will be expressed, and these other genes will not be expressed. So we wanted to apply this to these clonally expanded T cells. And yeah, as I mentioned, it's, this is rare, and it's only if it's very specific for infection. And so we don't want to just sequence, all of these T cells. You can see there are a bunch of blue ones, but there's still, the orange and the red, etc. But the orange and the red aren't necessarily what we're interested in. We're interested in these blue clonally expanded T cells. And so we are applying single-cell RNA sequencing, which is a new technology that is really gaining in frequency and has been helping in characterizing a lot of different diseases and development, etc. And so this is quite a straightforward concept. Single cell is going to be more informative than averages. So here's a smoothie, and here's some fruits. So if you take the smoothie, you don't know, is this piece, is it a blueberry or raspberry or blackberry? Rather, if you go in and have just the bowl of fruits, you can pick one up and figure out, oh, this is a blueberry, etc. And so to take this back to the T cells then, you can imagine if we take all of these T cells and look at the gene expression, maybe we have these two genes expressed. But what if it is that the blue ones are expressing this gene on the right, these other ones are expressing the gene on the left, and so, in fact, the average is that both are expressed. And so we apply this single-cell RNA sequencing. And so these next two slides are more data, so if you don't understand it, it's not much lost. I'll kind of tell you the main points. So this is a principal component analysis where we have sequenced T cells from healthy controls as well as CFS patients, and each point represents a single T cell, and they are placed on this principal component analysis plot based on their similarity. So if you have two T cells that are exhibiting very, very similar function, they will be placed very, very closely on this plot. And so I think maybe you don't understand the statistics, but if you look in red are CFS T cells, or T cells from a CFS patient. In blue are T cells from a healthy control. And you can see that there is a pretty clear difference between the CFS patient and the healthy control. And I don't show this plot, but we can actually go in and look at the genes that are contributing to these differences in the CFS and healthy control. However, the main point is that we do indeed see a difference in the CFS and healthy control. And so I've been talking a lot about these clonally expanded cells. So this plot doesn't represent that. This is just kind of a bunch of different T cells. So this next plot that I'll show you is a heat map showing the expression of different genes in a bunch of different cells. And so on the rows here are different genes. So these are genes, they have expressed RNA, they have different function. The columns are different cells. So we sequenced about 200 cells. This is all from a CFS patient. And roughly we've ordered these cells from left to right based on the extent that the cells have clonally expanded. And so, immunologists can go in and then look at these genes and kind of characterize what are these cells doing. And so there are a bunch of interesting genes, and they are basically as expected, actually. So you see a bunch of like granzyme B, granulysin, etc. These are genes that are actually responsible for killing the infected T cells. So just kind of to summarize this point, so I mentioned we did the single cell RNA sequencing. We were wondering, maybe these clonally expanded T cells have a different expression pattern as these unique T cells. And indeed, we do find that is the case. So these expanded T cells are expressing the gene on the right, for example, and these other ones not. And so basically what we find is that this gene on the right is responsible, or ones that are known to be responsible for removing infected cells. And so basically what we hypothesize then is that these in the CFS patients, we see an activated immune system, and this activated immune system is likely actually killing some cells. And so the summary of the overall talk, I think there are kind of two main points. The first is that the immune system is activated in CFS, and that this happens only in the event of a kind of specific recognition of an infection. And when you look at those cells that are activated, they do express the genes that you'd expect them to express, which are genes that are responsible for killing infected cells. And so, I think these are kind of preliminary results, but it does present a lot of, I think, hope and some things to follow up on. There are a lot of things that you can kind of correlate with CFS. We don't know, is it causing something? Is this, a secondary effect? One very powerful thing with immunology is, there is something that is activating the T cells, and then they are going out and killing infected cells. And so there is work going on in the Davis lab to actually characterize which, of this interaction with the infected cell and the T cell, what is stimulating these T cells. And so, I have been talking this whole time as in the context of an infection, but of course there's a lot of hypotheses that it could in fact be from yourself, so there could be an autoimmune disease. And so right now we have solid evidence that there is indeed activation. However, we don't know, is this response to an infection or is this response to self? And so this is exciting. However, we don't know, do you want to activate the immune system or do you want to inhibit it? And so these are things that we are looking into towards the future. And just the last slide, to thank especially the OMF for funding, and then all of the people who have done the work and given feedback. And thank you.